Hello, everybody. Welcome back, those who were in the morning session. As Mariana said, this is the second of two sessions today of our showcase webinars about our showcasing our Catalyst grants. As some of you may know, and maybe all of you, they were originally scheduled to be as part of our MICD annual symposium, which was planned for today and tomorrow. Those plans changed. We chose, however, to go ahead with the Catalyst Grant webinars because this is sort of the core mission and activities of MICD, and those activities are carrying on. So the plan always was to showcase some of the Catalyst grants that have been in progress over the past 3 years. We've had 3 years of this program so far, and we've drawn from Catalyst grant projects over these years. In this afternoon session, we have 3 Catalyst grant projects that come from the most recent cohort of Catalyst grant awards in 2019. We have the first presentation by Stephen Smith from Ecology and Evolutionary Biology, followed by Xuan Huan from Mechanical Engineering, and then Monica Valuri from Astronomy. As you may see even from the departments that the speakers represent, there is a diversity of research happening at University of Michigan in computational science, and MICD very much brings these to the fore and supports them, and we all learn from great, cool new things from them. So, okay, without further ado, I will hand the floor over to Stephen Smith. I will give you a heads up when you're about 20 minutes into the presentation, if that's okay. Try to do it as unobtrusively as possible, but I may have to turn my mic back on. And yes, if there are questions posted on the chat that seem like they should be answered right away, I will also interrupt and pose the question to you. If not, we'll go to them at the end. Okay?
Great.
All right, all yours then.
Thank you, and thank you for the introduction. So I'm going to talk today about biological complexity and evolutionary innovation, and in particular in plant genomes, and how the MICD Catalyst grant is facilitating some development of new methods there. So I am represented by that small little icon there, but then I want to highlight 2 graduate students that are working on this as well. Will Weaver at University of Michigan, EEB, a MICD student as well, and Nathaniel Walker-Hale, who's on the right there in the picture. He's at the University of Cambridge with Sam Brockington, but works with me and Sam on this stuff. Okay, so to start out, I just want to highlight some interesting facts, and I'm going to give a fair bit of introduction here because these topics are extremely diverse that MICD funds, and so I need to go in a bit more introduction than I might typically, but I will definitely get to the aims of the Catalyst grant. Okay, so you may or may not know, but there are 2 plant species that live in Antarctica. There's a grass species, and then this little thing on the right, Colobanthus. They are closely related to cacti. It's a little surprising considering where cacti live. Both of these are closely related to carnivorous plants. And all of these are closely related to beets. And all of this highlights that within this one plant group that has species that live from Antarctica to the deserts, that have interesting adaptations like carnivory, that exhibit this really interesting purple color that we see in beets, and that can be important crop plants. They include quinoa and things like that. There is something interesting about the evolutionary origins of some biological complexity and innovation that facilitates these plants to live in these diverse areas and have these adaptations. And so this is what drives some of this research that I'm going to talk about today. However, I need to give you a little bit of background because we're going to see a lot of phylogeny. And a phylogeny is the way by which we understand how lineages are related. So if we take humans, we might know that humans are most closely related to chimps. So we could draw this figure here. And at that point where they intersect, we understand that there was some putative ancestor that split millions of years ago. We also assume that there are extinct lineages along these lines that, Neanderthals, Australopithecus, and so on. In fact, actually, chimps are more closely related to another species called bonobo, but nevertheless, these are phylogenies, and this is how we understand this. Now, we can place this in a larger context, and humans, of course, fall within this larger group of mammals, the placental mammals. I'm pointing to basically where they are. And all of this, these phylogenetic trees, which for computational folks are directed acyclic graphs, are visual representations of the relationships between species and taxa, populations, and so on. So we're going to see a lot of those. They are quite an old concept. The first phylogenies, or phylogeny, is the only figure in Darwin's Origin of Species. In fact, he had it in his notebook 20 years before the Origin of Species. And these are foundational concepts for the biological sciences. So the tree of life, for example, is a representation of how all species are related. There are a few million species that we recognize now. We actually don't have one tree for all these species, but a fundamental goal is to build this tree and to understand the processes underlying it. And of course, to understand the diversity from things like this long-tailed widow bird to these crazy beetles that are quite scary, velvet worms, which are really interesting. We won't get into them, but look them up. They're cool. And of course, chimps and such. So they also help us understand many different processes and relationships in the biological world. So for example, they're used for studying cancer, infectious disease, Ebola, of course, the current infectious disease that we have. If you look at many papers on those, they will have phylogenies. We can also use them to understand dinosaur genomes or deer strains, etc. People also get them tattooed on themselves. Nevertheless, that gives us a little bit of an introduction to some ideas of complex traits and also phylogeny. So I'm going to give you a little bit of a bit more introduction to the kind of data that we're dealing with, and then talk about the methods that we're developing with this Catalyst grant. Okay, so we know that molecular data availability has increased dramatically over the last few decades. There are two ways in which this has increased. One way this has increased is in the number or the representation of species. So for example, if through time you made phylogenies of the available data for one gene in the public databases, if you made the available data phylogeny for 1996, you might have a tree that has a few hundred species. If you made it for 2001, it might have a couple thousand. And if you made it in 2011, it might have 20,000. So that's one way the data is increasing, is in the number of species. We can ask a bunch of different cool questions with this kind of data, or these kind of data. For example, did temperate plants adapt before or after they moved? So we think that temperate plants came mostly from tropical plants. And so there are adaptations like deciduousness, herbaceousness, things like that. So did they evolve deciduousness and then move, or did they move and then evolve deciduousness, for example? Turns out, there are a lot of exceptions, but for the most part, herbaceousness evolved before moving and deciduousness evolved after moving. Nevertheless, that's not the point, but we can ask a bunch of questions with these large species phylogenies. However, they are extremely limited for additional or detailed evolutionary inferences. For example, they don't have data for all the species, few genes are sampled very well, and genomic sequencing, which has become more common now, provides extensive more information for us to better understand evolutionary processes. And so to address the emergence of complexity and innovation across the tree of life, we need to use basically genomic data, which would be more gene regions for each species, in addition to the more species, to better understand things. So to give you an idea of what that looks like, this is the data for single gene regions that have been added to public databases. This is how much data has been added for genomes. So that blue line is the same blue line from the last plot. It's just that this red line, which is whole genomes, has completely eclipsed the blue line in terms of amount of data. And there are lots of projects that try and facilitate this kind of sequencing. For example, here's a 10,000 genome project that I and many other people are involved in to generate 10,000 plant genomes. Okay, so I'm going to introduce you to the study group that has primarily guided us a bit here. So the Caryophyllales is a group of plants, flowering plants, that has about 12,500 species and many families. And they are the species that I mentioned at the very beginning of the talk. So they include one of the 2 species in Antarctica, they include cacti, and so on. So they, as I said, include carnivorous plants, many of which you probably know, like the Venus flytrap or pitcher plants. They also, as I said, can exist not only in Antarctica, but in the highest mountains, Svalbard, all of these places. The places where you can find them are located on the right map there. As I said, they include cacti, which not only look really interesting, but also have interesting photosynthetic modifications that seem to have evolved in adaptation to those dry and warm environments. And they also have some really crazy morphological modifications, like that plant up in the top left, the Didieriaceae, looks like a Dr. Seuss plant. And then things like the Azoaceae, or the rock plants down at the bottom. Okay, so what drove some of the research for this, to at least gather the data, which occurred before the catalysis grant, was whether there are signatures of ecological and morphological shifts apparent in the genomes and transcriptomes. So the transcriptomes just meaning all the RNA. So there's a grant that was funded a few years ago to sequence a bunch of the transcriptomes and a couple whole genomes. We did this across the whole group. Here's just a general phylogeny of the group with where we sampled. And we get some general results. So for example, here is just a phylogeny, and every star on there is actually a whole genome duplication event. We won't go into detail, but suffice to say, we have found that within this group, there are tons of whole genome duplications. Here's a summary plot that somebody published a year and a half ago about where there are whole genome events, and you can see I pointed to the one in Caryophyllales that have 41 just within that one group. So we have suggested that perhaps these are linked with morphological innovation and such. To give you just an idea of that without going into too much detail, within the carnivorous groups, they're actually at the origin of most of the carnivorous plants, there are whole genome duplications that seem to be associated with that evolution of carnivory. But that's just one part of the story. So now we're getting to sort of the meat of what we want to better understand with our new methods. In addition to whole genome duplication, you can actually have single genes that duplicate. And these have been linked with several adaptations. For example, adaptations for Antarctic fish are linked to single gene duplications, as well as many of the things like, for example, the venom that is in duck-billed platypus has been linked to single gene duplications. We note also, in addition to whole genome duplications, single gene duplications within these groups, and some of them have been studied with more detail. So for example, there's a group, or what gives beets their purple color is something called betalains, which is an alternative to something called anthocyanins. Anthocyanins are what give pigment for most flowering plants across the world. Within the Caryophyllales, there is a switch from anthocyanin to betaline. They're exclusive to this group. They give slightly different colors, including that really intense violet that we see in beets. But they also have been of interest to biochemists and such as an antioxidant. And so we and others wanted to understand the evolution of this particular complex trait. And so when I say complex trait, I mean a trait that involves the evolution of multiple gene regions that impact some complex phenotypic traits, so some complex morphology. There are several questions that have long, pestered biologists for decades, which are, why do complex traits arise episodically? They seem to arise in these short time periods and bursts. Sometimes they're associated with more species, sometimes not. They often involve a complex of genetic elements and impact many parts of the phenotype. Nevertheless, they are distinct in that they are not sort of one gene, one trait. They're complicated things like this betaline pathway for this different kind of biochemical way of making pigments, or succulence, or carnivory. This plot at the bottom is just a way of showing genes through time that are linked in a network, and then some of those networks become stronger linked and then actually separate into different functions. And this might be one way that we think these complex traits can arise. So I'm not going to go into a lot of detail here, but I'm going to show, this is the part of the beta-lin pathway that has been worked out. And I'm just going to highlight these two genes. One is in red, that DOTA, and one is CYP76AD. So these are two genes that are essential for the expression of beta-lins. And these have been studied in more detail by our group and Sam Brockington in particular, the advisor of Nat Walker-Hale. And basically, what we've shown is that duplications in these two genes essentially facilitate the beta-lin pathway. So they are missing from taxa that have anthocyanin, and they are present in those that have beta-lins. And so there's been some some of this worked out where we know that CYP and DOTA duplicated at a particular point in time. And then later on, there was this co-option of those products that those genes have into the purple color that we know, the beta-lin pathway. A lot more of this has been worked out. So another gene actually facilitates this whole pathway to start and was duplicated several nodes before. But again, I don't want to go into too much more detail so that we can get to the methods that we have here. So what we've learned is that this beta-lin pathway from all of this kind of lab research has evolved multiple times within this clade, and is the result of multiple gene regions being duplicated. However, this is only part of this story, and it's a small part. So this is just the strongest signal. There are other genes and other traits that are linked to this. So this is where I get to what we've been working on. So there's a long intro to give you an idea. We are interested in complex traits and how they evolve, and whether we can identify the multiple gene regions and the multiple phenotypic traits that are associated with that, so we can better understand the evolution of complexity. So our three major goals are to detect shifts in evolutionary models, to expand all this beyond nucleotides, so to amino acids and other traits, and then to cluster different genes and phenotypic traits based on similar shifts in evolutionary models. So we've been working on 3 software projects developed as part of this. One is called Phiborg, one is called GoPhi, and one is Leaf Machine. Okay, so there are some new methods that we've been developing to detect heterogeneity. Our thought here is that when complex traits arise, there's new function, and it's going to show as a shift in basically evolutionary model. So one of those pieces of heterogeneity is topological heterogeneity. I'll show you a little bit of that in a second. And then another is model heterogeneity. So with topological heterogeneity, I just mean that you might have one gene, and it turns out that the gene might actually support different phylogenies in different parts of the gene. Here is a simple gene that shows in the beginning of the gene, it actually supports chimpanzee sister to humans. And at the end of the gene, it actually supports human sister to gorillas. Human sister gorillas. So we can have topological heterogeneity. So we have some methods to detect that. But we also, and more importantly, are developing methods to detect model heterogeneity. So most phylogenetic models assume a single underlying evolutionary model, a Markov model. However, there can be many shifts of this underlying the data, and current methods don't really accommodate for that. So this is work with Nat Walker-Hale. And basically, we've developed this algorithm where we can calculate the best model for each clade within a tree, sort by deviations from some underlying model, and then test adding this complexity, adding new models to this. And this is programmed in this in the software package GoPhi, which is it's parallelized, uses a worker pool design, scales really well to multi-core architecture, and it currently implements nucleotide models, models for morphological data, and some clustering of non-heterogeneous data. And we also include uncertainty, so we can understand where there are shifts in evolutionary model, as well as how confident we are in each shift. And just to give a couple preliminary results, we're very good at at false positives. We don't really detect false positives. When there's a single shift, we basically identify it 99% of the time correctly. 2 shifts, a little bit less accurately. 2 shifts where they're convergent, so 2 shifts in the tree that actually converge on the same model, we're pretty good at identifying. And then even if there are nested shifts within, so a shift and then a shift again, we actually are pretty good at doing that. Now, all of this relates to sensitivity. So the more different the model, this is based on Euclidean distances between models, the more different, the better we are at detecting it. So here, for example, is just a simple empirical example looking at one of the genes within that beta-lin pathway and showing that there's shifts in molecular model. Okay, so we're also expanding to other data types, and one of the most extensive data sets, most abundant data sets for plants is leaf data. And so with Will Weaver in EEB, he's developing this machine learning method to basically take leaf measurements, so then we can look at shifts in the evolutionary model of leaf evolution. So he's using herbarium specimens to basically, and then machine learning techniques to identify areas of these herbarium sheets where we can identify what are leaf material, what is stem material, what is the text of the herbarium sheet, and then of the leaves. So on the left, you can see some of those identifications, and then on the right, the green are leaves that are then going to be measured. And so we can use those data to then look at shifts in the morphological evolution across plants and how that might co-vary with different genetic molecular evolution. In the specific case of the beta-lin pathway, there's the expectation that the genes that code for beta-lin include things that increase tyrosine, an amino acid that is quite bitter and might be associated with herbivory. And there are many leaf morphological elements that are also associated with herbivory. And so we have an expectation that we're going to see shifts in leaf morphology that are associated with shifts in the evolutionary models of beta-lin development. And so we're using these machine learning techniques to be able to measure the leaves, which will then be used for detecting shifts in leaf evolutionary models that correspond to molecular evolutionary models. So the other software package that we're developing is called Fiborg, which is a mixture of some Golang and Python code that works with WorkQ out of Notre Dame. And basically this facilitates us to split up all of these computational jobs. In the case of these genomic resources, there are tens of thousands of genes, but each of our individual analyses actually requires heterogeneous resources. And so we can take advantage with Fiborg of the heterogeneous architecture of a bunch of different machines, including the cluster at the University of Michigan, as well as machines that are running in the lab, to sort of maximize our ability to analyze these really large data sets. And so Fiborg does that. So over the next few months...
Stephen, just to jump in.
Yeah.
Sorry, just to jump in. You're at 21 minutes, but looking good.
Okay, good, thanks. Yeah, so just a couple more slides here. So future directions over the next few months. So we're going to be incorporating the morphological measurements from the machine learning-based leaf measurement tool, so Leaf Machine, that Will Weaver is developing. And he is processing millions of herbarium records to basically get those leaf measurements so that then we can incorporate those into our evolutionary model analyses. And then with Nat Walker-Hale, we're developing the covariance tool set to cluster heterogeneous data. So we've already looked at clustering non-heterogeneous data, by which I mean if you had multiple genes that do not have shifts in evolutionary models, we can cluster those, and we're now developing that clustering tool sets for when there are shifts in those data sets, and we can then cluster based on those shifts. Those are things that we're looking forward to in the next couple months. All right, so just to wrap this up, I want to thank MICD, as well as Nat and Will, and then several of the other collaborators and funding sources that facilitate this. And then just to end with a nice picture of all the lab folks that I have. Okay, so thank you.
Great, thanks very much, Stephen. So I'd encourage the attendees to type in your questions in the Q&A box that you see. While waiting for that, waiting for them, maybe I can get things started off here by asking. So I guess at one level, I can understand, as an otherwise computational person, I can understand some of the models that go in here. I can certainly see a lot of classification-based methods, and I guess the CNNs also. I mean, you identify things and then you build classification models of that. So I'm curious to know more broadly, uh, since directed acyclic graphs underlie all of this, are those graphs used mainly for just as representation, or is there analysis also done on them using some of the tools that some of us may be familiar with in other from other areas? Not really tools, but other quantities that can be defined on these graphs, right? I imagine adjacency matrices are constructed and other things derived from that. I imagine centrality measures are important too, betweenness and things like that. I wonder if you could speak to some of that.
Yeah, so I would say that traditional graph summary techniques are actually less well utilized in phylogenetics or evolutionary biology than perhaps they should be, and that's primarily because of the way that we implement our models. So typically, our models are Markov models that use the tree to to basically estimate evolutionary distances, and so we typically develop more complex models instead of taking the final tree as sort of finished. So if that makes sense, we basically incorporate more complexity into our likelihood calculation, where we apply, where the tree functions as part of the model. There are newer techniques that are using basic graph summary statistics that you're mentioning to basically summarize the overall space that that's occupied by, for example, say a set of genomes. So, for example, you might have several genomes. You might construct phylogenies for every gene region that's within that genome, and you might end up with 10 or 20,000 different trees. And so people have been exploring graph-based summary statistics to summarize that whole space of all of the 10 to 20,000 trees, and basically try to interpret potential processes that might change that space. But that, I would say, is in its infancy right now. Yeah.
And something like a model shift that as you described it, is sort of central to your work. So how's something like that actually detected? I guess to a simplistic way of thinking about it is that, well, I could just traverse down a decision tree and perhaps detect a model shift. Is it? Could you say something about that?
Sure. So I would say I'll start in a different way. If you already knew that there was a shift, the way that it changes the way that you calculate the likelihood of seeing that data given the tree. And so you would apply one Markov model to part of the tree and one Markov model to the rest of the tree. So that's how you incorporate or calculate the likelihood if you already knew there was a shift. Now, how we detect that there's a shift is basically going through in a post-order traversal, and there's a minimum number of data points, or in this case, species, that are required to actually be able to have enough information to calculate the parameters of the model adequately. So given that minimum number, we post-order traverse through our tree and calculate the best model given the subtending data. And then in a very greedy way, primarily because of the size of many of these trees, which can be in the order of hundreds or thousands of tips, in a greedy way, we basically consider the strongest deviations to potentially be new model shifts. And then we use information criteria, so primarily Bayesian information criteria or Icaki corrected for size, AICC or BIC, to determine whether or not we should add that. And then once we have a configuration of shifts, then we can go back and look at the sensitivity and ask whether or not a shift that we've accepted is significant, that it should exist at all, and then whether or not that location of that shift is strong. So we actually perturb the area around it and test basically the neighborhood of nodes around it to see if we have confidence in the location of the shift. Yeah, hopefully that helps.
Yeah, no, thanks. That really helped.
Annette has a question. Oh, you have another question.
Yeah,
Sorry.
Oh. Hi, I'm Annette Osling. I'm one of the associate directors for MICDE. So just a quick question. I was wondering if there was one of your 3 goals that is like the most computationally challenging or some piece of it within there?
Yeah, I would say working out this algorithm to actually function was a bit challenging at first because basically the GoPhi set of packages that we wrote essentially have to re-implement a lot of phylogenetic analyses. So that was a bit of a challenge and took a lot of lead time just in generating the basic libraries necessary to do these calculations at a scale that would facilitate us to look at whole genomes and transcriptomes. Aside from that, the part that's managed by Phiborg, which is the distributed computing aspect, is probably the other one. So for, I guess to give some scale, so we're currently analyzing about 300 data sets, and then each of those, or 300 transcriptomes, and then each of those transcriptomes has about 25 to 40,000 genes. And so each, data set has to go through this pretty extensive set of tests for testing for multiple models. And so it needs to be done in a distributed way, but each calculation is actually in its own right a bit computationally intense. And so we had to figure out a way to scale that in a way that was a little bit out of the norm for a typical cluster run. And so we've managed to do that. The ones that we're looking for, the next challenges are actually going to be in that machine learning aspect. And so Will and I have been, he's been tackling some of the more basic machine learning, challenges, detecting clumps of leaves, for example. But then we've been, also trying to figure out the best way to scale this up, because as I mentioned in the talk, we have millions of herbarium specimens, and so anyway, that's what we'll be facing in the next few months.
And is there a particular problem you'll go after when you have it working more the way you want it?
Yeah, so there's 2. So one is going to be expanding, basically discovering the additional genes associated with the betalin development, but also, or betalin pathway, but also, as I mentioned there when talking about the leaves, we don't actually know what the selective pressure that drove the development of this pathway. We don't know what that was. There's speculation that it had something to do with herbivory and other things that we would expect to show up in morphological shifts. And some of them we can speculate on, but we're hoping that this will help us discover things that we weren't expecting before with that particular pathway. Another thing that we're going to be examining is we think that in doing some of our initial tests that we basically figured out while working on this particular grant, on the MICD grant, we discovered that we think that the shifts in evolutionary model might actually correspond to life history. So things that grow more quickly, so herbaceous plants, or say rodents versus elephants or something, things that grow more quickly, they seem to exhibit stronger shifts in the rates. And so we're going to be exploring that as well.
Cool.
That's all my questions for now. Thanks.
Okay. Thanks for that. Any more questions, maybe from some of the other moderators? If not, okay. Thanks very much, Stephen. We will move on to our next presentation. Thank you.